Hello, this is the Epic's Urban Farming Prototype Team. We have a partnership with Peace Garden and Farms and Pastor Marty Henderson in Gary, Indiana. We are working to fight food insecurity through urban farming. Currently, Gary, Indiana is a food desert in which residents must travel over a mile to the nearest local grocery store. With the help of a design of an aquaponics system, we will be able to provide fresh produce at a subsidized rate to residents. This study determines the symbiotic relationship of fish and plants in a stacked system through aquaponics. To define aquaponics is the symbiotic relationship between fish and plants in a single ecosystem. Through this ecosystem, the fish will provide excrement and nutrients to the plants, while the plants will purify the water for the fish. The objective of this research was to determine the compatibility of two species in a stacked aquaponics system. The reason for choosing a stacked aquaponics system is that people will be able to grow multiple beds of plants within a single square foot area, compared to your traditional aquaponics system where you can only go one bed in the same amount of area. So our system will be growing 3 stacks of pea microgreens with a 200-gallon tank of bluegill. The reason for choosing pea microgreens is this is a fast-growing plant in that they are pretty easy to take care of once they are finished growing. The reason for choosing bluegill was this is what our stakeholder wanted, since it is a traditional fish eaten in Gary, Indiana.
We decided that we would grow 17 pea microgreens in our prototype and 1 fully grown bluegill in a separate tank. Our prototype has a constantly running filtration system that circulates both water and nutrients throughout the entire system. The bluegill was fed daily with fish pellets, and once a week we would add fish water from the tank into our prototype system with the plants. While growing the plants in the system, we kept track of the concentrations of ammonia, nitrite, and nitrate daily with a kit we have. We also monitored the pH and adjusted the water to balance our pH to where we needed it to be. Every time we checked the concentration and pH, we would also measure for plant height.
The results of our project, and basically we were able to get the peas germinated initially once we switched the germination process to be done in sand, and then we had initial high pH values in the water that we had to counteract, so we initially added vinegar to counteract those, but we realized that once the plants would be put in the system, that vinegar would not be allowed as it would harm the plants. So that's when we knew we had to switch to another source. So we got a fish tank with fish water with natural microbes creating and the biological, all the additives being created. So once we add the fish water, the pH leveled out better. There was initially a large ammonia amount, but the microbes and everything in the plants and the actual system were able to convert it to nitrite and nitrate. And at that point, the plants began to grow better, and a lot of the pH and concentration measurements we were having issues with initially were starting to level off. And once the plant's growth became more normalized and more average after it got out of that first high grow period, the pH concentrations and the concentrations of the ammonia, nitrite, and phosphate all leveled off.
In the upcoming fall semester, we plan on continuing our research, but this time we'll be increasing both populations. The pea microgreens will be doubled to 34 plants, and the bluegill will be increased to 10 bluegill. Thank you for taking the time and to listening to our research presentation.